to Extreme Lifeforms, the podcast where we dive into the weirdest, most incredible organisms on Earth. I'm your host, Maria Mamontov, and today we have a very special guest, a psychrophile, an organism that thrives in extreme cold. Please welcome Psychromonas ingrahamii. Thank you for having me. Can I just say the temperature in your studio is quite uncomfortably warm for me? Oh, I apologize. I can change the temperature for you. What is your ideal temperature? I thrive at a colder temperature around 5 degrees Celsius. That's my most ideal temperature for growth. But I am comfortable from anywhere between 10 degrees Celsius to negative 12 degrees Celsius. Although it's below the freezing point of water, where most organisms usually require liquid for parts to move around, bacteria, I don't become frozen solid. Wow, that's incredible. Do you prefer extreme cold or can you just tolerate it? The cold is my happy place. Unfortunately, other bacteria can barely endure the cold. I function and grow best when it's cold. If the heater is on 20 degrees Celsius, I will struggle to survive. I grow with generation time of 240 hours at negative 12 degrees Celsius and can even grow at lower temperatures. It is the lowest recorded growth of any organism verified by a growth curve. How fascinating! Could you introduce yourself to our listeners? Where do you come from and what role do you play in our environment? Well, I originally was discovered in 2006 by a team led by Berkermans Nielsen's. I was named for John L. Ingram, and I was isolated from Sea Ice Point Barrow in Alaska, USA. I hang out in the cold marine environments. I benefit my environment by playing a role in biogeochemical cycles in frozen environments, and I also help break down organic matter in cold ecosystems. Thank you. So, what makes you special? In what ways are you exactly extreme? Well, I am an extremophile, specifically a psychrophile. A psychrophile means is someone who is loving the cold, usually a bacteria or archaea. I am a bacteria that not only survives but thrives in extreme cold temperatures. I am one of the most cold tolerant bacteria known. I am capable of surviving in environments even colder than permafrost. Like I've mentioned, you'd find me hanging out in Arctic or Antarctic, permafrost, sea ice, and more. What's interesting about me is I hold the record for the largest number of gas vessels among known bacteria. What makes you well adapted to these freezing conditions? Well, the extremophilic adaptations that I produce are antifreeze proteins from the gas vessels that help me survive in extreme habitat, maintain my ice crystal formation, and protects my cell structures, which ensures proper function and makes sure I can float in icy waters, allowing my cells to remain near nutrient rich surface layers instead of sinking into deep nutrient areas. My cell morphology, large, rod shaped cells with some distinct adaptations for freezing conditions. I also produce enzymes called psychrozymes that have evolved to function efficiently in low temperatures. These enzymes have a looser, more flexible structure than those found in mesophilic organisms, allowing them to work efficiently in cold without needing a lot of energy. At freezing temperatures, most cell membranes would become rigid and non functional. I deal with this by adding unsaturated fatty acids to my membrane, keeping it flexible in the cold. These fatty acids help lower membrane's freezing point, making sure nutrients and waste can be moved properly. Unbelievable. What do you eat to survive? I feast on organic material trapped in ice or floating in the ocean. Even in the cold, microbes like me are essential for breaking down nutrients in our ecosystems. Can you talk a bit more about your metabolic and genetic traits? Absolutely. Because I take about 10 days to grow, this means that I have a slow but efficient metabolism because I conserve energy, operating well with minimal resources. I am aerobic, which means I need oxygen for survival. That's why my home is extremely important to me. Alright, thanks for that. Now, how do you connect to the field of astrobiology? What a cool thing about me is I grow and survive in extreme conditions and temperatures that resemble a planet like Mars, for example. That's why I'm considered a model organism for astrobiology. Because of my ability to survive in extreme cold, I've suggested that there may be life on icy moons like Europa and Enceladus, where living species can survive like liquid water under ice. Really? But how would you be able to survive on the moons if you need oxygen to survive? Great question. I probably wouldn't be able to survive. I need oxygen, and Europa's ocean has almost none. But if I could adapt, new traits would favor using like sulfates or iron instead of oxygen. Europa's seafloors might have hydrothermal activity, providing alternative energy sources. Plus, my gas vessels might not work under pressure, under high pressure. So I'd need to evolve. That's so cool. Follow up question: how do you connect with learning about life beyond Earth? Well, it suggests that other living organisms might be able to survive somewhere beyond Earth. I am a great extremophile that expands the understanding of habitable zones. Scientists understand how life can survive in frozen extraterrestrial environments. If microbes like me can adapt to Earth's harshest environments, why not somewhere else? Overall, I provide insight into potential biosignatures on other planets. That's mind blowing thoughts. Before we wrap things up, do you have any final words? I hope you keep looking for life in unexpected places because life is absolutely adaptable and you never know where you might find it. Thank you for having me. Thank you for joining us today. I'm your host, Maria Mamontov, and thank you for tuning in to Extreme Life Forms.